In Chapter 2, we are going to explore protein composition and structure. So some of the goals that we have for this chapter, first, to understand the different levels of protein structure, know the different amino acids and their classifications. So we'll look at the functional groups that are present for each of the amino acids, and we will classify these into groups like polar or nonpolar. We want to understand the acid-base properties of amino acids. So here we'll look at things like the pKa. We'll explore that in a little more detail. And we want to understand primary structure, which is that first level of structure, and how peptide bonds form. So how do we hold this primary structure together? We also want to know and understand the different types. So we have different types of secondary structure that exists. More specifically, we have alpha helices and beta sheets. We want to understand tertiary and quaternary structure and how these are different from one another. We want to understand how the amino acid sequence will impact the folding of the protein, and how to differentiate between different types of proteins. And this will be based on the shape and solubility. So here what we're looking at is, we have globular proteins versus fibrous proteins. So what are proteins and why are they important for us? Well, proteins, they are linear polymers, okay? And these polymers are built of individual units, these monomer units, which are amino acids. So we have individual amino acids that have certain functionality with them that come together to form an overall polymer structure. Now, because we have different amino acid groups that are present, we have a wide range of functional groups present in our overall protein structure. Because of the functional groups that are present, these proteins are able to interact with other biological macromolecules, so we can form more complex assemblies. So proteins, they can interact with other proteins, they can interact with DNA, lots of possibilities for these interactions based on intermolecular forces. And we do know that some proteins are rather flexible, but some proteins are going to be quite rigid in terms of their shape. So in this figure here, we are looking at proteins we have different subunits of proteins here that are interacting with one another. We have proteins that are interacting with DNA. So this is, in a lot of ways, highlighting all of those properties that we just listed. So this really allows us to see the complexity of proteins. So here what we're looking at is we have these large polymers, and these large polymers are going to be made up of individual amino acids. And they start off as a nice long chain of amino acids, but because of the functional groups and because of the side chains present there, they're able to form intermolecular interactions and kind of have short range and long range attractions within this one polymer unit here. They're also able to associate with other units, and these functional groups can associate with some of the functional groups that are present in the DNA molecules. So we can see that other biomolecules will interact with proteins. Now, the structure of the protein is going to be important, and we know this because we've heard that structure ultimately dictates function. So structure dictates function, and the protein's primary structure will be based on the sequence of the linked amino acids that are present. Then you go on to the secondary structure, which is really the three-dimensional structure that's formed through intermolecular forces. Then there's the level of tertiary structure, where we have longer range intermolecular forces that are forming more complex three-dimensional structures. And then quaternary structures, where you have interactions of different polypeptide chains or different protein chains with one another. And we'll explore all of these as we move through this chapter. So we've mentioned these different side chains, these different amino acids which make up proteins, and we've talked about how they're really going to give some functionality here. And these functional groups which impart that functionality or allow for different types of interactions include the following molecules. We can see alcohols present in our protein structures, thiols, thioethers, carboxylic acids, carboxymides, and a variety of different basic groups are present. So lots of functionality here, and this is going to rely on you going back to your organic chemistry and thinking about what these functional groups actually represent. So remember that in alcohol, we have some R group with a hydroxyl group. Thiols, we have some R group, and then we have a sulfur and a hydrogen. The thioether, we have an R group, a sulfur, and another R group. For our carboxylic acids, you usually have some sort of R group here, and then we have that COOH present in that structure. And the carboxymides, which will also be called aminocarbonyls, are going to have some R group connected to that C double bond O. Which is then attached to that nitrogen and two additional R groups present. And then, of course, the variety of different basic groups. We'll classify these as we move through. Now, again, to show the complexity and the importance of different proteins that we have, this is looking at, we have this figure here where we're looking at different interactions or larger interactions between protein structures. So we know that proteins can interact with one another, and they can interact with larger biological macromolecules. So this figure is representing an example of a complex protein assembly. So here what we're looking at is a single muscle cell which contains those myofibrils, and myofibrils, they're comprised of numerous repeats of protein assembly within them. Now, another example, which is not shown here, but another example of these types of interactions would be the interaction of proteins that are involved in the DNA replication process. Some of the protein structures that we look at are going to be very rigid. Now, substances that will have very rigid structures, these are proteins that are going to serve as structural elements. And so the rigid proteins are going to be present in the cytoskeleton or connective tissue. Okay, these are structural elements. But we also have some proteins that have a little bit of flexibility, and because of the flexibility, they're going to act as maybe hinges or springs. A lot of times when we're talking about proteins, and we'll see when we talk about protein regulation, proteins actually have different conformational states, or they undergo conformational changes when something binds to it. And this is a common means of protein regulation. So here, what we're looking at is a substance that the unbound state has one conformation, and the bound state has a different conformation. And so we're able to differentiate between the unbound and the bound state. And in this case, this is a protein, it's the protein lactoferrin, and it shows how the binding of just a little iron within this pocket is going to significantly change the structure of the protein. So now let's get to those building blocks of these protein structures, the amino acids. Now, amino acids will exhibit some special chemical features. These will include the capacity, what's this here? the capacity to polymerize. They also have some novel acid-base properties. They have varied structure and functionality. And they also have chirality. The general basic amino acid structure, we have a central carbon, which we call the alpha carbon, labeled here. From the alpha carbon, we see that we have an attached amino group, carboxylic acid group, hydrogen, and then our R group, which is our variable side chain. And the nature of that side chain is going to determine how we classify this amino acid. In terms of chirality, amino acids, remember chiral molecules, we have non-superimposable mirror images, and we're going to have different forms here. So we will use the D and L form, and the assignment is based on how the enantiomer rotates plane polarized light. The L isomer is going to rotate counterclockwise, and the D will rotate clockwise. But we will use the D and L definitions when we talk about our isomers. So, as an aside, you've seen the S&R system in classes, so you've probably used the S&R system in organic chemistry, and that's based on the idea of prioritizing the different side groups that are present. Where the highest atomic number gets the highest priority, the H or hydrogen atom would get the lowest priority and point away from you. And then you're looking at whether you have clockwise or counterclockwise. So the S version is counterclockwise based on priority, which means then that our R is going to be clockwise in terms of priority. Now, again, we use the D and L version instead of the S and R system. For almost all L forms of amino acids, they actually exist as the S isomer as well. When looking at our amino acids in terms of their acid and base properties, we can think about the functional groups that are present here on these structures. Now, we do have two functional groups within the amino acid structure, at least two, this amino group and this carboxylic acid group, which can either have a proton present or have a proton removed from them. And where that proton is removed is based on the pKa value. And so at different pHs, we're going to have different versions of our amino acids. So at neutral pH, which is what we're talking about when we have physiological conditions, physiological pH, you end up with your amino acids present as dipolar ions. Okay. When you have this dipolar ion, we call that the Zwitter ion. So we see here in this figure, this is our pH scale. At lower pHs, both of these groups have hydrogens present. When the hydrogen is present, the amino group is positively charged, and this carboxylic acid group has no charge. Well, as we're increasing the pH to this range of physiological pHs here, that carboxylic acid portion loses that proton and becomes negatively charged. So this is the Zwitter ion that's present. Now, if we increase the pH even further, then you'd see that the other group, that amino group, would lose its proton, and that would lose its positive charge. And so this structure over here is no longer that Zwitter ion. We will revisit this idea as we continue to talk about classification of amino acids. So the categories of amino acids that we'll look at, we have hydrophobic, polar, positively charged, and negatively charged. So hydrophobic, think about these as nonpolar substances. So hydrophobic have the nonpolar R groups, and there are actually 9 different nonpolar or hydrophobic amino acids. The polar will have polar R groups, and there are 6 of these. And then the positively charged have a positive charge at physiological pH. Okay. And there are 3 of these. And then our negatively charged then would have a negative charge at physiological pH. And there are 2 of these. And so we're going to look at the overall structures of these substances. Starting here with our hydrophobic. So we have glycine, alanine, proline, valine, leucine, isoleucine, methionine, tryptophan, phenylalanine. Those are our 9 different hydrophobic amino acids. Now, you will need to know the structures of the different amino acids and how they're classified. So you do want to start putting together some flashcards so that you can see the differences here. And so, for example, with glycine, which is a simple amino acid, we see that the R group is just a hydrogen that's present there. When we move to alanine, that R group is a CH3. So that R group is changing as we're moving through our structures. In terms of the polar amino acids, we said there were 6 of those. We have serine, threonine, tyrosine, asparagine, glutamine, and cysteine. And again, you need to know the structure. So they have the same basic structure, but it's our R group that's changing in these cases. Our R group is going to be different from one amino acid to the next. Our positively charged, we have lysine, arginine, and histidine. And then our negatively charged, aspartate and glutamate. Now, in addition, the amino acids, we do have abbreviations that we use for those when we're just showing different portions or segments of a polypeptide chain if we're not drawing out the structures. So each amino acid, just general notation, each amino acid has both a 3-letter abbreviation and a 1-letter abbreviation. Now, more often than not, the 3-letter abbreviation kind of makes sense based on the name of the amino acid. The 1-letter abbreviation doesn't necessarily relate directly to what that amino acid name is. But oftentimes you will see throughout this particular textbook, rather than listing or drawing the individual structures out, they will give you a sequence that looks something like this. And so what that means is you start with an alanine, and then you have a leucine, and then you have your tryptophan present here. But that just represents what that overall structure would be. So we talked about these Zwitter ions that are present, and we talked about how that formed because we have 2 groups that are present, both that amino group and that carboxylic acid group, which can gain or lose a hydrogen. But there are several amino acids that have side chains. In addition to those 2, that amino group and that carboxylic acid group, they have side chains that are also able to be readily ionized. So here we're looking at a list of some of these substances. Within this list, we do see there's our carboxyl group and our amino group. These are the groups that we've already talked about. But we also see that aspartic acid and glutamic acid, aspartate and glutamate, have a side chain that can lose a hydrogen. We have the histidine, cysteine, tyrosine, lysine, arginine. All of these are side chains that are ionizable. And so when that proton is removed from the side chain, that's dictated by the pKa. And so we can see how this varies depending on what that side chain is. So here's a nice table that kind of lets us look at the pKa values of some more common amino acids. Here's the amino acid listed here. This is the pKa value for that carboxylic acid, for the deprotonation of that. And here is that amino group. And then those that have ionizable side chains are present here. Now, we can look at these and see that there are some common trends with the carboxyl group and the amino group. And so the average pKa of this carboxyl group is 2.9. That's the average. And the average pKa of the amino group is 9.5. So they're very similar in terms of their numbers for both the carboxylic acid and the amino group. Now we can't really group those ionizable side chains together. They're very different from one another depending on what's present. So what this tells us, this pKa value is going to tell us approximately what the pH is where that proton is going to be removed. So as we're approaching values of about 3, you're going to see that for these amino acids, these common amino acids listed, really for all amino acids, that carboxyl group is deprotonated. And then when you get to pHs that are around a 9.5, you're going to see that amino group has lost that. But remember, we said that in between at physiological pHs, because of these two values, at physiological pHs, we end up with that Zwitter ion because this would be negatively charged and this is still positively charged. We haven't deprotonated it yet. And again, some of these, because of the side chain, will have some impact on the charge that's present depending on the pH. Now we do go through and we explore this a little bit more when we're talking about our laboratory experiments. So again, just to reiterate, we have these Zwitter ions that form. You have at physiological pHs, we end up with that carboxyl group losing the proton and becoming negatively charged, but our amino group has not yet lost its proton, so it's still positively charged, so we end up with a Zwitter ion that's present. Now we can explore or look at titrations of amino acids. So this is showing the titration of alanine, and what that basically is telling us is where are we going to end up losing those protons, or what are those pKa values. So this is just pulled from another textbook to kind of point out what we can do with these things. And so, you see kind of at those leveling off points or in between there, you see where you are going to have your deprotonation occurring. And the number of humps in the titration curve, remember, is based on how many ionizable protons do you actually have. So something like histidine that has that ionizable side chain is going to have a more complex titration curve. And you can see a leveling off for each of those protons that's going to be removed. Now when we talk about the amino acids, we talk about the 20 amino acids which are really relevant for living systems. And we've looked at how we classify these things and the functional groups and how they change in these. But we do have other amino acids that exist. So why is it that these 20 amino acids are the amino acids that actually become incorporated into biological systems? Why are some of the other ones not? Well, part of that is because we've seen that there's a lot of chemical versatility with those 20 amino acids that we already have. They also are the amino acids that were present in these prebiotic reactions. Remember that whole idea of the primordial soup and the formation of life from that. And there's also a problem where some of the larger amino acids that exist outside of this list of 20 may be too reactive. And so we don't want those in biological systems because we don't want all of these side reactions taking place. So now that we've looked at the amino acids and their chemical versatility and their side chains and their properties, let's start to get into the overall structure of proteins, bringing these amino acids together. So the primary structure, or the beginning level of structure that we have, this is going to be based on the amino acid sequence itself. Okay. So with this amino acid sequence, we are forming these linear polymers, and these polymers are formed because when these amino acids come together, they form a very specific type of bond called a peptide bond. When that peptide bond forms, it's accompanied by the loss of a water molecule. We saw this in chapter 1 when I just briefly showed you the polymerization of these larger biological macromolecules. And we saw in all of the cases that water was lost in the process of the polymerization. So here we're showing this peptide bond formation. We have two individual amino acids which are going to come together. So the carboxyl end of one is going to bond to the amino end of another. And when that happens, we're going to lose water. So an oxygen is going to come off of here, and two of the hydrogens are going to be removed here. So our oxygen and two of the hydrogens will be lost as water. And we're forming this peptide bond between the carbon and the nitrogen. And there's our water given off. And so every subsequent amino acid that comes in is going to form this type of bond. So you'd have another amino group coming in and interacting with this carboxyl group. Now, peptide bond, we actually have resonance structure that's present here. It results in double bond characteristic, and that's important because it limits the rotation about that peptide bond. So the peptide bond itself isn't a double bond there, but because of the resonance that we can have, you end up with partial characteristic. It helps stabilize that bond a bit, provides some rigidity to that. So we're limiting the rotation. Now, when we're looking at peptide bonds, we can think of these in terms of planar structures. So for a pair of linked amino acids via that peptide bond formation, there are actually 6 atoms which are going to lie within this plane. Okay, so every time you see that peptide bond, planar structures. In terms of the bond length that's associated with these, remember that here is our peptide bond formation. So about 1.32 angstroms, that bond there. This was our bond between our alpha carbon and our carboxylic acid carbon here. And then our amino group and our alpha carbon here. Okay, so this is just showing some typical bond lengths that are present. Now both cis and trans forms are possible when this peptide bond forms. In the trans form, the 2 alpha carbons are on opposite sides. Okay, so you see here alpha carbons on opposite sides of that kind of plane. In the cis form, they're going to be on the same side. So there's our cis form in the same side. Now the trans form does tend to be favored over the cis form, and that's because the trans form is going to minimize steric hindrance. So while both forms are possible, the trans form will be preferred. So we know that the peptide bond is rather rigid, but the other bonds that are present within our polypeptide chain, they are going to have more rotation. They will rotate more freely. This is going to allow the structure to fold in different ways because we know we have this long polypeptide chain, but then we know different groups that are present are going to interact with one another. So we have to be able to bend these. So there are some key terms that are associated with bond rotation, and these terms include the torsion angle, phi, and psi. The torsion angle refers to rotation about bonds other than the peptide bond. The phi, which is represented by this symbol here, this is going to describe rotation about that N alpha carbon bond. And then our psi is going to give us information about our alpha carbon to that carbon from our carboxylic acid bond. Now the phi and psi angles will ultimately determine the path of the chain that we have. We use something called a Ramachandran plot to allow us to view the different angles of phi and psi and the likelihood as to whether or not those angles are going to be possible. So the plot allows you to view allowed angles of phi and psi. So the allowed values are going to be the more favorable angles, and those are going to be shown by the dark green. So dark green on here is going to mean more favorable. So in the picture, the angle combination shown in the lower right quadrant, so the combinations that are shown here, which we see in this structure, are going to be unlikely. And that's because we see that there's steric hindrance that's present. Whereas those angles that are shown in this upper left quadrant are going to be more likely because we minimize that steric hindrance. So the angles that we observe are going to dictate kind of the way that these are going to fold, but the polypeptide, even when not considering these angles, does have directionality that's present. And that's because we have these different ends. That's because we have the amino and that carboxylic acid end. So we have what's referred to as an amino terminal residue, which we'll see on the left, and that carboxyl terminal residue, which we'll see on the right of the structure. So these, because of these different ends, it imparts directionality to the overall structure. So we bring a bunch of these amino acids together, form these peptide bonds. We have long polypeptide chains. Most polypeptide chains will be between 50 and 200 amino acids in length. Of course, we have some that are shorter than that and some that are smaller than that. Those with a small number of amino acids are typically referred to as oligopeptides. Okay, so these have smaller numbers of amino acids together. Those that have larger amino acids are called polypeptides. The polypeptide chain contains a backbone, and then from that backbone, so here's our backbone going through, from that backbone we have these side chains, these variable side chains that come off of that. The hydrogen attached to the nitrogen is a good hydrogen bond donor. So that's our good hydrogen bond donor. And the oxygen of the carboxylic acid is a good hydrogen bond acceptor. And this is going to allow for the interactions between some of these substances that will help us start to form our more complex structures of proteins. Now, we can, within these structures, because of those side groups that are present, we can end up with something called cross-linking. This will occur in some cases. The most common substance for cross-linking will occur with disulfide bonds. So a disulfide bond is the most common cross-link observed in proteins. Cross-linking results from the oxidation of those cysteine residues. So if you have cysteine residues that are present in various points within a structure or various points between structures, you can end up having oxidation of those cysteine residues, and then those cysteine residues can interact with one another. So what we're seeing here is in chain A, we have some cross-linking between cysteines that are not next to one another, but that are close to one another in the structure. And then two different chains, chain A and B, cysteine residues from each of those can cross-link with one another. So this is allowing us to see how complex the different types of interactions are within these structures that will help us get towards those higher order structures. So why do we care about the amino acid sequence? Well, remember that whole idea of the central dogma where our DNA to RNA to proteins. Okay. Remember that the sequence that we end up having is going to dictate the shape, and the shape is going to dictate function. So the amino acid sequence is determined by the nucleotide sequence of the genes. So the sequence is ultimately determined by the DNA. And we know that the DNA codes for the RNA. And the RNA specifies the amino acids and how they're going to be put together. And we'll look at that through the course of this semester. When we have alterations in the sequence, so when there is some mutation that occurs, that ends up leading to an abnormal structure, which leads to abnormal function. And that's when we're going to start to see different disease, because these proteins are not functioning properly because they don't have the correct structure. So we've looked at primary structure and that sequence. Now we're getting into the secondary structure of proteins. Now the secondary structure will result from the regular patterns of hydrogen bonding. So here we're looking at more localized intermolecular interactions, and specifically hydrogen bonding between these substances. And there are two main types of secondary structure. We have our alpha helix and our beta sheets. So starting with the alpha helix is a coiled structure which is stabilized by that hydrogen bonding. So there's some terminology that we have that's related to this alpha helix structure. When we're describing the alpha helix, we can talk about the screw sense of the alpha helix. And the screw sense tells us the direction of rotation. This can be right or left-handed. A right-handed is clockwise. And then our left-handed would be counterclockwise. The right-handed, or the clockwise, is going to be the more energetically favorable form because there are fewer steric clashes when we have that right-handed form. So when we see the alpha helix in nature, in proteins in nature, we tend to see nearly all of the alpha helices in the proteins in their right-handed form. Now the alpha helix content can vary from protein to protein. Some proteins will have no alpha helices, and others will be almost all alpha helices. So we can vary between 0% helix and 100% helix, with varying degrees in between. Now when we're looking at the general structure and what's present here, the alpha helix, we have 3.6 residues per turn. When we're saying residue, what we're talking about is the amino acid that's present there. We have what's called a pitch of 5.4 angstroms. And when we're talking about the pitch, it's the distance, if we're looking at, say, the structure here, the distance between the green balls on one side of the structure. So here to here. Okay, that's our pitch. So we can describe in a little bit more detail what's going on with this structure. So hydrogen bonding will stabilize the alpha helix structure. The carboxyl, that C double bond O that's present of each amino acid will form a hydrogen bond with that amino group, that NH group that's present, and it forms it with the residue or the amino acid that's 4 residues away. So the H bond forms between that CO and an NH that is 4 residues away. So we're starting with this one. We're counting 4 residues. 1, 2, 3. Here's our 4th residue. So the hydrogen bond is going to occur between these two substances. All of the main chain C double bond O and NH groups are going to be hydrogen bonded in these alpha helical structures. So residues are described in terms of the rise or the translation that's present. And the rise is 1.5 angstroms along the axis of the helix. And each rotation is 100 degrees. Now that rotation, that 100 degrees, represents 3.6 residues per turn. The pitch, which we talked about, that distance between those, will represent 1 complete turn. And that pitch is equal to the rise times the residues per turn. So that, for example, that 5.4 that we talked about for that pitch, we said that the rise was 1.5 angstroms, and we said that that rotation was 3.6 residues. So that gives, if we have the pitch being equal to rise times residues, that will give us a value of 5.4. Now the other secondary structure, these beta sheets, these are 2 or more strands that are interacting with one another, rather than one strand forming a tight coil. The strands that are interacting with one another are going to be hydrogen bonded together. In a nearly fully extended beta sheet or beta strand, the distance between the adjacent amino acids is about 3.5 angstroms. So what we're talking about there is the distance between the neighboring amino acids within that. When you have it in the helical portion, because they're tightly coiling, that distance is much shorter, or the adjacent amino acids are much closer to one another. Now, in the beta sheets, we actually have 3 different types of orientations that the beta sheets will fall into. These beta sheets can be considered anti-parallel, parallel, or mixed. The anti-parallel, these are going to be sheets that run in opposite directions. The parallel will run in the same direction. And then the mixed has strands that are parallel and strands that are anti-parallel. And we can see this in this figure here. So the first portion, these are strands running in opposite directions. So this is going to represent our anti-parallel. Here we have strands running in the same direction. So these are going to be our parallel. And then in the bottom picture here, we see that some are going this way and some are going this way. So this is going to represent our mixed. Now when we do have, so the anti-parallel versus the parallel, you see that the bonding, the hydrogen bonding, when you have these parallel structures, will end up being shifted or distorted relative to that hydrogen bonding that we see in the anti-parallel. Now we won't focus too much on this, but there are what are referred to as supersecondary structures that exist. So these supersecondary structures are taking these beta sheets and these alpha helices and putting them together in different ways. And so this is just showing us a couple of different ways that we can combine these structures. So in the top here, we have a beta alpha beta unit. So this is our beta, alpha, beta. That's what the secondary structure is referred to. So it's two beta portions that are kind of linked by an alpha helices. Here we have an alpha-alpha structure. This is referred to as a beta meander. And then this structure down here is what's called a Greek key. So again, just different ways that these alpha helices and beta sheets can come together. And you'll see in all of these cases that we have little linkers that occur between the different motifs, between the different structures. There are some other observed structures that we come in contact with. We will see coiled helices, beta barrels, zinc fingers, and leucine zippers. So these are all additional structures that we'll see. So this is just a side-by-side looking at a protein that is mainly alpha helical in nature and one that is mainly beta sheets in nature, just so you can see the differences in the overall structure between these substances. I did point out when we were looking at the different types of structures coming together that we will have some little connector pieces that occur between different structures. So some of these are referred to as reverse turns and loops. So loops allow for more compact structures, and loops will often be referred to as omega loops. They're more elaborate in structure. They have no regular periodic pattern to them. It's just kind of the bending over of that portion. Reverse turns, we can also see beta turns or hairpin turns. They involve that carboxylic group, that C double bond O. That residue is hydrogen bonded to an amino group residue. And it's the carboxylic acid group of 1, which we'll call the I residue here, is going to be in these reverse turns, is going to be hydrogen bonded to the amino group residue that's 3 away. So it's the I plus 3 residue. And this is going to help stabilize the overall structure. Now the loops and turns will tend to lie on the surface of the protein, not embedded within the core, but rather at the surface of the proteins. Now we talked about helical structures, and we do have a couple of special types of helices that we encounter when we're looking at protein structure. Alpha keratin and collagen are two special types of helices. Alpha keratin, this top structure here, is comprised of two right-handed alpha helices, and these two right-handed helices are intertwined. They come together, and when they come together, they're going to form a left-handed superhelix. Left-handed superhelix. So we refer to this as an alpha helical coiled coil. And then collagen, this is comprised of 3 helical chains that are intertwined with one another. And so we see the 3 different chains are labeled with different colors here. The structure within the collagen is going to be stabilized by steric repulsion. And this forms what we refer to as a superhelical cable. Superhelical cable. All right, so moving beyond secondary structure, we're getting into the tertiary structure that's present. So we said primary structure was the sequence of the amino acids. Secondary structure, we're looking at closer hydrogen bonding between residues that are near one another. Now with the tertiary structure, we're starting to look at longer range interactions between substances. so they're not as close to one another. And this is going to help us see more complex folding within the protein structure. So here what we're looking at is the structure on the left here. This is myoglobin. The structure on the right, we're looking at a porin. This is just to show us different types of proteins, and both of these are globular proteins. Globular, they're kind of in the shape of a glob. So the folding in these, usually what happens is we have the residues coming together based on their properties. And so hydrophobic or nonpolar residues will tend to want to interact with other nonpolar, and polar residues will want to tend to interact with other polar substances. And so oftentimes we end up seeing that our nonpolar residues will end up forming interactions within the inside of the overall structure. Okay, so they kind of embed themselves within. And the more polar interactions, which is represented by the blue here, tend to be on the outside of the structure where they can interact with, say, water. Now, porin is kind of a special type of protein that forms a channel. So in the porin structure, because we're forming these channels that water can move through, you're going to end up with the polar on the inside and partially on the outside of these structures. But we do see here that the polar will tend to interact with the polar and the nonpolar with the nonpolar, based on the types of intermolecular forces that we've talked about both in Chapter 1 and through more extensive study in general chemistry. Now, as these proteins are coming together and forming these larger interactions, I think it is important to note that there are differences between those supersecondary structures that we've observed and these overall larger compact globular units. So we refer to as supersecondary structures, we refer to these as motifs, whereas these compact globular units that are coming together, we refer to as domains within the overall structure. So remember the supersecondary structure that one example is, in this case, we have an alpha turn alpha or helix turn helix. So, in some proteins, you're going to end up with these repeating regular structures that are present, and others, you just have compact units coming together. So they're just folding into these compact regions that don't have any regularity that's associated with them necessarily. All right, so I'm moving from tertiary structure to the quaternary structure. Here what we're looking at is taking these polypeptide chains, or these linked amino acids that have folded into three-dimensional structures, and now taking two different polypeptide chains and interacting them with one another. So in the tertiary structure, we're forming three-dimensional structures with long-range interactions within a polypeptide unit. In the quaternary structure, we're taking two different polypeptide units and allowing them to interact with one another. So here we're looking at hemoglobin as an example of this quaternary structure. So hemoglobin, you have these four different chains that are coming together, so four polypeptide chains, and the chains themselves are held together through intermolecular interactions. This is not one big chain that has folded in on itself. These are four individual chains that are coming together to interact with one another. So quaternary structure is looking at multi-subunit structures, interactions between polypeptide chains. So we've explored the different levels of protein structure. But how do we determine the structure of proteins? Well, in order to figure out the structure, if we're looking at the primary structure, or if we want to know the primary structure, we have to go through and denature the three-dimensional structure that we have. So how do we denature the three-dimensional structure? Well, we do this using various chemicals. And so some of the chemicals are listed here: urea, guanidinium chloride, beta-mercaptoethanol. Okay. These are substances that are going to help break down these three-dimensional structures. The urea and the guanidinium chloride. So these two structures, or these two chemicals, are going to disrupt non-covalent bonds. So these are disrupting those intermolecular interactions. And then this beta-mercaptoethanol, this will allow us to break disulfide bonds. So interactions between those cysteine residues. So this is just showing that breaking of the disulfide bonds. So we'd use beta-mercaptoethanol. So you have your overall large structure, and you see these are places where we have disulfide bonds that are present there. And so these chemical substances are going to come in and break apart these bonds that are between these sulfurs. So we're reducing the disulfide bond. And in doing that, as the disulfide bonds are reduced, that beta-mercaptoethanol is going to be oxidized. So being able to go through the process of taking this three-dimensional structure and breaking it down, that allows us to get information on the amino acid sequence or that primary structure, and then that's going to allow us to learn about protein folding. Now, once we have denatured a structure, it is possible to have refolding of proteins. Sometimes the refolding will occur, but you have to have suitable conditions in order for this to occur. So sometimes it occurs on its own if the conditions are right, but sometimes we're going to require what's called chaperone in order to aid in that folding process. And so those chaperones are going to be molecules that help kind of direct the refolding of these proteins. So throughout this chapter, we've looked at what amino acids are and how they lead to proteins and the different levels of protein structure. So you will want to go back and review those amino acids. What are the amino acids? What are the side chains? How do we classify them? How is the primary, secondary, tertiary, and quaternary structure formed from these overall amino acid sequences? What does it mean to be a Zwitter ion? Why is that important or significant? And think about, while we didn't intentionally say it, think about how modifying that side chain will impact the type of intermolecular interaction that you have present.